Hey, how's it going, sir? So this is the video for question 1, explaining the DNA replication process. So this first page I have is just an example of what DNA would usually look like. So you have the double helix over here, you have the coil, and then you have the corresponding base pairs or nucleotide base pairs with their hydrogen bonds. So dark green and regular green are adenine and thymine, and orange and yellow are guanine and cytosine. So adenine and guanine, or adenine and thymine, both have two hydrogen bonds keeping them together, whereas guanine and cytosine have 3 hydrogen bonds together. I also included these little blue dots as deoxyribose sugars, and or brown dots as phosphate groups. The top strand is labeled 3' to 5', and the bottom strand is labeled 5' to 3'. Okay. So the process is going to start with DNA helicase, moving through the strand, unwinding it, and unbinding the hydrogen bonds between the base pairs. And that's going to create something like this, where the hydrogen bond, or with the nucleotide base pairs, are now separated. To keep them separated, these things called single-stranded binding proteins, which are these purple little circles, they're going to bind to each strand to prevent hydrogen bonds from binding to each nucleotide. Okay. And you're also going to have topoisomerase or gyarase, which is going to be on the DNA strand, just relieving any tension from the unwinding. Okay, so you can see here, again, purple circles are the single-stranded bonding proteins, and this like blue coil, which is this thing, is going to be the topoisomerase, just relieving any tension from the unwinding. Okay, and after this, we're going to get over to the elongation part, which is going to be represented in this page. So it's not really that well done, but I did my best. Right, so as soon as the 3' end of the parent strand opens, so this being the parent strand, this the top one, replication of this strand begins, and this is going to be the leading strand. So this is going to be from 5' to 3' now, right? To start this elongation, RNA primase, which is this thing right here, it's going to put a primer right about there to start the process and to allow DNA polymerase 3 to create additional nucleotides along this new strand, right? So this little red circle is DNA polymerase 3, and this is the RNA primer, right? This is RNA primase, that's the primer. Okay. Now onto the bottom strand, which is going 3' to 5'. You're going to see these little fragments right in between each strand, and that's because DNA polymerase 3 has to go in the direction of 3 to 5, or 5 to 3'. And since this one is going 3 to 5', RNA primase has to set up primers at every 5' region, which is going the opposite way. So what's going to happen is the primer is going to be set at 5, and then DNA polymerase 3 is going to work backwards, right? And then once that strand is filled, RNA primase has to come back and set up a new primer at another 5', and then DNA polymerase 3 is going to work backwards again, and then so on. And then you're going to be left with these Okazaki fragments, and those are going to be filled up with ligase right here, right? Eventually, the DNA elongation is going to stop, and to fill these fragments, we're going to need DNA ligase, which is going to act as a glue and fuse the two gaps together, or the two sides together, to make one strong DNA strand. And we're also going to have to remove the DNA primers or the RNA primers and change them into DNA nucleotides. So to do that, you're going to need DNA polymerase, and that's going to go, all through the bottom strand or the lagging strand, and also at the top strand, which is the leading strand. And it's going to just convert these into regular DNA nucleotides, just to balance out the new DNA double helix. So in the end, once again, RNA primer, so the red part, and DNA ligase, which is the glue, kind of conjoining the Okazaki fragments together. So in the end, this is what it's going to look like, and you're going to have 2 equal pieces of DNA, or 2 equal double helixes. So yeah, that's pretty much it. I think I touched everything. And yeah, so also because of how this part is behind or does develop slower than the top region, because of the 5-3 elongation process, the top one is called leading strand, and the bottom one is the lagging strand. I think I didn't touch on that before, but yeah, that's pretty much it. So at the end, this is what it should look like. Okay, thanks.